Greetings, my name is Peter Flint, and this is my project for BIOLG8490 entitled The Efficacy of Various Acaricides to Combat Mites in Honeybee Population. On the left-hand side here, you see a picture of Apis mellifera, the common honeybee of choice for apiarists around the world. On the right-hand side, you see the parasitic Varroa destructor, Varroa mite, which is infecting honeybee populations around the world. Here's the article and the source material, and the impact factor is 2.21. A little bit of background on the whole research project, and it starts with the life cycle of the mite. Varroa mites affect bee populations worldwide. In the early 1900s, they jumped from the Japanese Asian bee, Apis cerana, to Apis mellifera, which now means they're affecting the population of bees which are chosen for honey production around the world. First, initially, the queen will lay the egg. As she lays an egg in the brood chambers, the worker bees will come along and they'll tend to that newly formed larvae. In this case here, you can notice a small mite on this worker bee, and what happens is the mites are blind and deaf, but they have a perception and a sense of smell that allows them to realize they're over a larvae. They jump off and they kind of tuck themselves underneath the larvae, which are hidden. Some hygienic species of bees will pull the larvae out and throw them away. In this case here, it's hidden. The bee will cap the brood chamber cell, leaving inside the developing larvae and the mite. The mite will go through some reproductive stages, produce more, burrow into the side of the larvae, and start to digest some of the valuable nutritional material that's used by the larvae to develop, including fat bodies and hemolymph. Sadly, eventually the bee will hatch, but it's hatching in a weakened state because some of its nutritional material is taken away during the developmental stage by these parasitic mites. This is problematic because if this continues without being treated and goes on for 3 to 5 years, something called colony collapse disorder, which is very descriptive in exactly what it is. A colony collapses due to the number of mite population being so great that it overwhelms the bees. The other thing that happens is the bees become weakened and are susceptible to different viruses. One of those would be deformed wing virus, which is attached to the number of mites that are in a population. So it's important for apiarists around the world to control the number of mites and keep that number down and the bee number up. One of the ways they do that is through what are called acaricides. Acaricides are insecticides aimed specifically at these mites. They tend to be plant oils, essential oils, or some sort of plant derivative. Because keep in mind, you want to kill the mite but not the bee. A couple common ones would be like thymol, which we're familiar with as the active ingredient like Vicks Vapor Rub. It's a strong, pungent smell. That's effective at killing mites and not the bees. Another will be oxalic acid, which is if you ever run through stinging nettles, you get that acid underneath your skin. It has that irritating effect. Well, that is not as harmful to the bees as it is to the mites, so oxalic acid becomes another one that's used pretty regularly. The research in this case here used 5 different acaricides: Apivar, Apistan, Hopguard, which incidentally is attached to the hops for beer. The essential oil pulled out of the beer hops is an effective acaricide. Checkmite and Apiguard. And these are all readily available products to beekeepers, which they can basically mail order, put them in their hive, and try to keep the mite population down. It's important to note that prior to the test, beekeepers in this research project would count the number of mites. They would do it during the test treatment and following the test treatment to get some good numbers to work with. And they also used a control group throughout the process. It's also interesting to note that there are 2 cases where they used a half dose and tested for the efficacy of the half dose. And the reason for that is the mites do build up a measure of resistance to some of these chemicals, so to test a half dose to determine the efficacy of that would be wise and help them realize, hey, if a half dose works just as good as a full dose, why not use the half dose? Here's the first experimental process. This will be classified as a laboratory process in the case of the research. What they did is they built 8 boxes and 5 sets of those for n equals 40, where they had a queen and a screen bottom, a nutritional source, the pollen patty, a sugar source, the syrup, water, and then in the middle here the treatment strip. So this was carried across 8, and you notice down here the control has no treatment, and the different types of treatment processes are used: Checkmite, Checkmite half dose, Apiguard, Hopguard, half dose. Again, the mites were mite counts were made before, during, and after application, whether that was counting on the number of mites that fell through the little screen here and they died, and And the researchers would count the number of mites on that screen, or they use what's called a sugar shake method, which basically, as comical as it sounds, it actually is pretty effective and used by beekeepers all around the world. You take a container, you put a bunch of bees in it, you put some powdered sugar in there, and then you shake the container with the bees in it. They have a hard exoskeleton, so it doesn't damage them. The mites fall off the bee. You put the bees back in the hive. And the sugar that's all over them, it's like being coated in their own food. They eat it off, and it's okay. But in the container now, you have a mixture of mites and sugar. The sugar is water soluble. You add water, and the reddish-brown mites show up in contrast against that clear solution. You're able to count the mites very easily, and those are the numbers that they're working with. Same thing on the experimental group called the field group. This is where we had 5 Langstroth hives, a typical wooden framed honeybee hive, times 4. We had Apiguard, Apistand, Apivar, Hopguard in the control group with no treatment. And keep in mind here, and now by the way, the n equals 20 in that case, keep in mind you have your sugar shake. So here's the before, no treatment, and the sugar shake test was done to get a mite count. Now the acaricides are applied. So this, and they're following that, they have another sugar shake. So after the acaricide is applied, there's a test run to get a number there. And then finally, after they do the final removal, a test is done. So you have 3 tests here, the before, the during, and the after, to get the numbers that you want to work with. And again, a control group is present and the sugar shake method was used. What were the results? Well, here's really what we want to see. A high number of Varroa mites killed and a low number of bees killed. And that was found in Apivar. So a high number of mites were killed here, but bees were not. And the same is the case for Apistand and for Apiguard. What was noticeable though is the half doses. If you look at the Hopguard had a large number of bees that were killed and also a large number of mites. But when you cut that in half, the research found that the Hopguard was effective. So a half dose of that was good, and a half dose of the check mite was good as well, as opposed to using the full dose. Here you have a pretty substantial number of bees being killed, but also a large number of mites. So it's effective, but not nearly as effective as you've cut the dose down. What happened when the comparison was made between the boxed hives out in the field and the laboratory boxes? The researchers indicate there really wasn't that much difference between these salmon-colored cage bars and the teal-colored hive bars. In fact, the comment, they said the mortality level of the mites was about the same between both groups. A couple conclusions and discussions. First of all, it became apparent to the researchers that it is important to monitor your mite population over the time. You can't just simply put in a Apivar strip and then walk away from your hive thinking there it's effective and it works the best. You have to do the before, during, and after. In fact, the research showed that about every month you should check the mite population and insert a Apivar strip as needed, because it was obvious from the research that initial doses of the acaricides had a really substantial effect right off the bat. Within 6 hours, massive numbers of mites were dying. However, that tapered off quickly and they resurged, so it was important to continually check and continually treat the mites. These are the sources that were used, both outside from this article and others outside of it. I found this to be an incredibly helpful article that I really enjoyed. And in fact, we have some modular units that we're having to build for my class, my AP Bio class, because of the COVID situation. So this is going to be one of my go-to articles for that, and helping the students realize the scientific process, the scientific experimental method, and how data and research drive some of the decisions. So I plan on using this article for that module. But if you have questions on it, I'd be happy to answer those. Thank you for your time.